So, actually, the title of my talk is only provocative. So it would be a nice discussion for a workshop, but I don't think we have time to go deep into this question. But while I will be discussing the work of two of my students, Federico Gherardini and Francesca Sacco, I will try to make the point that we should use this technology with some care. So, since we had this problem, I forgot to thank the organizer and the committee for inviting me here. And I really enjoyed the meeting. I've seen quite a lot of nice posters, nice presentations. And most of you address very interesting biological problem using a solid reductionist single few genes approach. And this is good. But probably you already experienced the strange feeling when you start reading the literature that your favorite protein, especially if it's a functional protein, is involved in a very complex network. And you have sometimes you have difficulty to try to predict what is the result, the outcome of an experiment if you have to consider all these complex circuits. So it's not your fault, it's not the fault of your brain, it's actually it's an important problem. So to do that, you need to model your system and come up with predictive model that can help you in designing and interpreting experiment. So I think from what I hear from your talks and from the presentation, most of you have already had experience of having to cope with larger, noisy data set and try to extract useful information. You can do this with some experience with a spreadsheet program like Excel, but if things become a bit more complicated, then you need something more, and perhaps you need the help of an expert, an bioinformatician colleague. So what I'll try to do in my talk is to start with some general consideration of holistic approaches. And then I'll be dealing with two general strategies that together with Federico and Francesca we developed with the goal of developing a system to map cell perturbations to specific pathway on a large scale. And then the goal of identifying direct targets of signal modulator. This type of approach can be classified in the journal term of systems biology that have become rather popular recently. Because if you look at the production, if you do a Medline search with a string system biology, you see that there is an exponential growth of abstracts that contain this term. But what is more important that also editor of fashionable journal look with some sympathy to this type of manuscript, because if you now calculate the total impact factor of this manuscript, the growth is much steeper. So I spent some time before last week to come up with a clear definition of systems biology. But then after some effort, I resort to just going to some systems biology journal, look at the abstract, and see what this paper, this manuscript were describing. So essentially, there are two categories of approaches that we classify as systems biology. One is genome-wide data gathering. To have a paper with a system biology protocol, we need to sequence a genome, produce a transcriptome, better more than one transcriptome, some interactome, metabolome, epigenome. And then another characteristic of system biology manuscript is modeling. So you have to come up with some model of your system, and this model needs to be predictive. So do we need more genome-wide data? The simple answer is yes. But we also have to consider that there are already a lot of data in databases. And you probably know this journal. Some of you might have also published in this journal. And probably you have seen that a few weeks ago there was an entire issue dedicated to the UNCODE project. So this is a collaborative project of a large number of groups, and what they have done is to map signals on the genome level on a large number of genomes of approximately 300 cell lines coming from different tissues. So this is the outcome. I mean, I couldn't put it on a slide. I'm sorry for those of you in the back line. They might not be able to see that in front of this gene promoter there is a lysine 3 methylated on lysine 7 on histone 3, and so on and so forth. I mean, these are only 2 genes, and you have a mapping of all histone modification, DNA sensitivity, promoter bound by transcription sites, and so on and so forth. So if I wanted to bring the whole data with me, I would need a poster that is 30 kilometers long and 16 meters wide. But fortunately, informatics people invented databases, so we don't need to have all the data on a poster, and you can go to your favorite gene in the ENCODE database and look where there are modified histone, which transcription factors bind to your favorite gene. But I think I also invite you to try to think about all this data and see whether you have a good idea to look them with a holistic view. I mean, not just your favorite gene, but at the level of gene regulation, the level of whole genome. It's not only ENCODE, because, for instance, you have heard about the 1,000 genome project, so 1,000 genome sequenced. Then there are at least 2 projects, one at Sanger Center and one at Baylor College, in which a large number of tumor cell lines have been sequenced. And the genetic variation in a number of coding sequences have been correlated with resistance to tumor. The HAP-MAP project. So there are a lot of data there, and as Andrea Ballabio has shown to us yesterday, it's worth having a look at this with hypothesis, of course, before starting proposing to do a new transcriptome or a new sequence, a new gene. So another point of system approaches is modeling. So let's try a simple experiment. So a simple linear pathway that most of you know, are familiar with, only 5 proteins, and there is a linear relationship of activation between proteins. So without any doubt, all of you will be able to tell me whether MAP kinase is activated if I add the EGF to the system. Of course, you're right, all the system is activated. It's slightly more complicated if now add some more relationship. For instance, a feedback loop between MAP kinase and the source protein. Probably we need to think a bit longer, but most of you will be able to tell me that we have to consider this pathway in time, because first MAP kinase will be activated, but once the feedback loop is activated, the MAP kinase will sense the negative feedback and will be turned down. Again, slightly more complicated, we add some more modifier interactor. EGF receptor is phosphorylating GAP1, and this promotes the attachment of RAS, or RAS gap that inactivates RAS, but SHP2 is able to inhibit the GAP1 by the phosphorylating it. And perhaps, I mean, we don't have time now to go into the detail, but this is still something that we can solve by thinking. But then if we do the experiment, we go to the literature or pathway databases like KEGG or Reactome, and we extract all the information that we have about logical relationship between proteins that have been described in the literature that are likely to affect this pathway, we come up with this. And now I challenge all the important professors in the first lab, in the first seat, to tell me what happens if I add EGF and then I inhibit this node here and predict whether the cell will grow or go into apoptosis. I think we cannot do that. So we need a model. There are different types of models. And also, I think I have only to describe them with name. Boolean model, Bayesian, fuzzy logic, ordinal differential equation. There are different level of regularity. They can, sorry, granularity, not regularity. They can describe the system in different level of details. And the ordinal differential equation are the one that more deeply can describe on the chemical level the system. But of course they cannot handle large network. If I want to handle a network like this, I have to use much simpler model like Boolean model, where I don't have quantitative, not input quantitative data, but just on and off relationship. You don't have to be scared about the name, Boolean, Bayesian, fuzzy logic, because there are good tools, simple tools available on the web that you can use by clicking here and there. So think about considering to use this modeling approach and have a model of your favorite system. So now, after this simple introduction about what we mean for systems biology, I'd like to describe what I'm going to do next, what are our interests. Essentially, we would like to try to link molecular information to phenotypes through this black box or gray box here. Because what we would like to do is to see how the system can be perturbed either genetically, chemically, or artificially via saRNA, and how these affect high-level phenotypes, like for instance diseases. So this is not very innovative because Thomas Hunt Morgan more than 100 years ago had similar goals. What is different, and now we have quite a lot of information about this black or gray box, and there are in the literature, in database, there are a lot of information about logical relationships between proteins. So as I showed before, we have, now we can model the cell via complex network. If you take all the information that has been annotated in the Reacton or KEGG databases, and you plot in a single network, then you come up with a complex network of approximately 3,000 proteins linked by a large number of edges. So I'm going to describe soon the experiment done by the project of Francesca Sacco, and in order to avoid confusing you, I want to point out that when we are be talking about modeling from now on, I'll be considering only Boolean models, where each node of the protein defined in my system can be either on or off. And I'd like to try to be able to address this following question. So I'd like to predict the phenotypic effects of perturbing the activity of any node in this model. So for instance, if I activate A, it's not very difficult to see here that C will be activated, D will be on, while E and F will be down. So this is one first goal. The second goal is I would like to be able to predict the molecular targets of any perturbation producing a phenotypic effect. So I take my cell, my system, I do something to the cell, either add a growth factor or perturb with a chemical, and what I observe that the node that I'm measuring change their status. So can I, in some way, by combining experiment and modeling, map this perturbation on one of these nodes? And by little thinking, we can see that by this simple experiment, the predicted target of the perturbation is node C. So Francesca Sacco decided to tackle this type of problems by looking at a family of proteins called phosphatases. So as you know, signaling is mediated by a large number of modifications. Probably the best known, I don't know whether we can say the most important, is phosphorylation. And phosphorylation, as you all know, is mediated by a class of enzyme called kinase, many of which have been characterized as oncogene, whose function is that of adding a phosphate group to either tyrosine, serine, or threonine. Conversely, there is another large family of protein called phosphatase that remove this phosphate. And also, phosphatase have been considered boring enzyme. Not as boring as lysozyme proteins, but close. And so if you, again, if you do a bibliometric search and you look at the number of papers, you see that kinase here in red have received much more attention than phosphatase of the past years. And again, the thing becomes more dramatic if you look at total impact factor. So in green is the total impact factor of phosphatase protein against kinase. So the advice for the student here, if you want to do a productive PhD on and get paid for total impact factor, don't work on phosphatase, work on kinase. But I did this type of analysis only recently. Francesca didn't know that, so I convinced her to do a PhD on phosphatase. So phosphatase is a large family of proteins, approximately 300 proteins if we also consider regulatory subunit of phosphatases. And as I said, they've considered till recently, of course I'm a bit exaggerating now, phosphatase is, the intrinsic phosphatase is growing in recent years, but until now were not considered as sexy as kinases. So Francesca, with the help of Federico, and also I made some contribution, decided to adopt this four-step strategy. First, assemble a cell-specific model. We'll try to see what's important, this term cell-specific. Define readouts, experimental readouts that can be used to characterize the cell state. Then measure the cell state, measure this readout in a large number of perturbation states. And finally, map the perturbation to the model and identify which one is the node on the model that sends the perturbation. So this is the prior model, the literature model that Francesca drew by simply looking at the literature. You cannot probably read it, I cannot read it here on my computer, but it's a rather complex model, and probably it's too complex. You don't want to treat such a complex model. But you have to consider that in doing this, Francesca took experimental relationship derived from different experimental systems, different cell lines. We don't have evidence that all these connections are working, are acting in a cell system that is actually HeLa cells in this case. So we want to optimize the model. We want to have a large number of experiments in which we measure the largest possible number of nodes, the activity of largest possible number of nodes, and train the model. So use some algorithm that changes a bit the model and selects the model that best explains the result. So to do this, you need to do what is called a multiparametric analysis. So in each cell, you measure the largest possible number of parameters. So Francesca did 7, and she measured these 7 parameters in, I can't remember how many now, but approximately 50 to 100 conditions. So in 50 to 100 different conditions, we know whether ERK is activated, P38 is down-regulated, AKT is up or down. So this is what's called a poor man multiparametric analysis, because there are better way to do that, but we don't have access to instrumentation to do this. For instance, by directed proteomics, you can do much better, and there are very interesting new instruments coming out with single cell analysis or multiparametric analysis. But still, this was a very heroic effort, and finally it paid off. So essentially, under each experimental condition, each perturbation, we measure 7 parameters, here only you get 5, and we identify the status of the system by what we call a cell state vector that tells me what is the activity of each of these nodes under that condition. So the cell state is defined by this vector. So by using a software called Cell Nectar Optimizer that was developed by Julio Rodriguez, that is now in Cambridge, we were able to train the model with experimental results. So essentially we move, we delete edges and nodes, and we explore a large number of conditions until we find a cell-specific model that exactly or best reproduce our experimental result. So essentially in this table, in this color code, you see the training data, the experimental result, where red means upregulated and green downregulated. And here, in this other table, you see what the model predicts in this condition. And here is the difference. Green means there is no difference, while red, that is absent, would mean there is a lot of difference. So this diffuse green color tells us that our model is good in predicting the experimental results. So now we have a model, and it was developed and trained on specific data sets, but we can use it to predict experimental conditions that have not been used to train the result, to train the model itself. So for instance, we can ask what happens if we activate this node, what's the PI3 kinase or whatever, or AKT, and then in this case we let the model predict the outcome, and we do it in any possible up or down regulation condition, and we come up with a table like this, where we have now cell state vectors that tells us what happens when we upregulate AKT, or we not downregulate AKT, or ERK, or GRAD2, and so on and so forth. So these are predicted outcome of the experiment. So now we want to use this system to map perturbation on the model caused by phosphatases. So we have 300 phosphatases, we have to do a lot of experiments. So what we do, what Francesca did, is to have high content siRNA screening by using automated fluorescent microscopy. So essentially we have a lab tech slide, microscope slide, where we spot 300 siRNA against each of the phosphatase, and then we plate the cells on this slide, and by automated fluorescent microscopy, we measure the activation of a number of readouts, what we call the sentinel proteins that define the state of the system. So in this experiment, she measured NFKB migration to the nucleus, P38 activation, ERK activation, ribosomal protein S6 phosphorylation, and appearance of LC3 dots to monitor autophagy. So since as all high throughput experiment have problem, what she did is to use 3 different siRNA for each phosphatase to avoid off-target effects, and each experiment was repeated in average over 3 different biological replicates. So now if we look at specific sentinel protein, like for instance S6 phosphorylation, and we look at the distribution of signals in the 200,000 different images. By the way, I forgot to tell you that we don't look at the images one by one, but we treat them with a software that is called Cell Profiler, that automatically look at the images and determine the intensity and the size and the quality of the fluorescent signal. So essentially, if we look at this signal and we draw a distribution curve, we see that quite a lot of experiments have a signal that is not distinguishable from the unperturbed cell. But then there are a number of experiments that are at the tail of this curve that show a variation of signal. You might be able to see here that with a scrambled siRNA, if you look at 6 phosphorylation, you see some signals here and there. But then if you inactivate this regulatory subunit of PP1, that's called PPP1R3C, you see that the signal is down-regulated. So this means that this subunit is a positive regulator of this sentinel protein, while if you inactivate this other, the catalytic subunit of the PP3 phosphatase, then the signal is very much increased. So this is a negative regulator of S6 phosphorylation. If we look conversely to LC3 dots, we see that in the scramble vector there are a few dots. If we inactivate PTPN21, the number of dots decreases, while if we inactivated this other subunit of PP1, then we see an increase in the signal. So by this type of approach, we identify what we call hits. Hits are experiments where the signal is statistically very different from the one obtained with the scramble siRNA control. So we represent the data in this way, where square green represents phenotype, molecular phenotype, S6 phosphorylation, NFKB migration to the nucleus, and so on and so forth. While circle represents phosphatase. So if we find that the phosphatase, inactivation of a phosphatase, affect the phenotype, then we link the phosphatase to the phenotype. Red means that this phosphatase is predicted to be a negative regulator of this phenotype, while green means a positive regulator. So we see that not only phosphatase, I mean, this is nothing particularly new or innovative, but not only phosphatases are not only down-regulator of growth signal, but some of them are also positive regulators. So now, how much of what is known in the literature is reproduced by our experiment? So essentially 15% of the hits have been clearly described in the literature. I have to admit that there are quite a number of relationships that have been described in the literature that are not reproduced by us. And there are a variety of reasons why that doesn't happen. Either the siRNA didn't work well, or we made some mistake, and so on and so forth. Of the remaining 75% of the hits that are novel, these were confirmed by secondary screening with a different siRNA library. So one observation is that there are many phosphatases that influence more than one phenotype. For instance, this one is actually is perturbing 4 different phenotypes. So there are 2 possibilities to explain this. Either the phosphatase that we are considering as multiple substrate in this library, this is possible, or alternatively the complexity of the circuitry or of the network is such that by hitting a single substrate, then you influence the whole cell state by changing the output of more than one sentinel protein. So, to explain what we are going to do next, I'd like to lead you through this type of reasoning. So suppose that we have a phosphatase that perturbs NFKB migration to the nucleus. This doesn't mean that NFKB is the primary target of this phosphatase, because we will obtain the same result by inhibiting P38 or IKK or ERK. But since we have this multiparametric analysis, we don't have just one output. We have the chance of mapping with more detail, more precision the perturbation. Because now, if we know that this phosphatase not only activates NFKB, but also ERK, then we know for sure that the target of the phosphatase is not the IKK P38, but must be ERK or upstream signal. So essentially, we use this type of reasoning to map phosphatase on the model. We have 2 different cell state vectors. One is the experimental cell state vector, the one that we obtain by multiparametric analysis by siRNA screening. So for instance, we know that by doing a siRNA of the phosphate dose 1, we activate LC3, but we downregulate RP6. And then we have our model that predicts for each perturbation of the nodes. Nodes, remember, do not contain the phosphatase yet. Our goal is to map the phosphatase on the model. So by perturbing each node of the phosphatase of the model, we know what is the predicted cell state. And now what we do, we conclude that the experimentally observed effect of inhibiting a phosphatase is the same as regulating the model, the node in the model here, then the phosphatase is predicted to be an inhibitor of that node. So we compare cell state vector, experimental one after inhibiting phosphatase, and the cell state vector after informatic inhibition of the node. And by doing this, we were able to map, if you remember well, 43 phosphatase on the model. And I want to tell you that this node do not represent the substrate of the phosphatase. They represent what we call the entry node. So the node in the model that first sends the perturbation due to inhibition of overproduction or overproduction of that phosphatase. 15 phosphatase were not mapped on the model since no inferred profile, the one predicted model, could match the one that we obtained experimentally. So the explanation of this lack of success could be due to the fact that either the phosphatase have multiple targets or that simply our model is not completely correct. So as you can see here, this blue or yellow dot means that these phosphatases have been described in the literature as oncosuppressor or oncogenes. And by this approach, we were able to map more precisely what is the likely target of their activity. The one that had not been characterized earlier as oncogene or suppressor have been tested by Francesca on a simple assay looking at cell cycle timing by looking essentially at the Ki-67 proliferative antigen. And what she was able to show that these phosphatases interfere with this network, also interfere with cell cycle timing. So now a bit of validation. So essentially, can our mapping, our model, predict experiments that have not been used to train the model? So essentially, let's focus on these 4 phosphatases. I don't know if you can read them on the screen, but the name doesn't really matter, because 2 of them have been mapped on the receptor TNF-alpha, while the other 2 have been mapped downstream of ERK. So now this mapping predicts that perturbing or either by cRNA or by overexpression the 4 phosphatase, this will have a different outcome on the activity of RAF, because the perturbation of the phosphatase that has been marked on TNF-alpha will have an effect on RAF, while the one that downstream should. And I will tell you if this will turn out to be true, and as Francesca was able to show by this type of experiment. So the first conclusion of this part, we developed a novel strategy whereby computational model of cell signaling are used to map on a large scale results of perturbation screenings. And by this approach, we were able to map 43 of the 58 hit phosphatases on 10 model entry points. I've shown that the predictions can be validated experimentally, and one of the conclusions that most phosphatases do not have redundant function, because if it were true that phosphatase, there are 2 or 3 different phosphatases that perform the same function, we wouldn't have been able to do our screening. So the fact that we find hits means that many phosphatases do not have a redundant function. So, but now this is still a low resolution mapping, because we map the phosphatase on a target on the model, but as I told you, this is not the subset of phosphatase. Can we go one step further? And this was done by looking again at this simple pathway, linear pathway, the RAS pathway, considered linear in this cartoon. And as you know, perturbation of this pathway, either in somatic cell or in germline cell, can produce a number of genetic diseases or cancer. So, but many of the patients that show this Noonan syndrome, Leopard syndrome, or Costello syndrome have mutation in a single gene called SHP2, one of the phosphatases. But still, there are quite a number of candidate genes that need to be discovered, because there are a number of patients whose mutation have not been mapped. And so the problem is can we find a modulator of that pathway that I've shown to you before, so that we can identify new candidate genes. And then secondly, some of the patients carrying the same mutation show different phenotypes, showing that the genetic context is important. So this is the motivation. I'm going a bit more deeply in this. So I want to show you the case of two phosphatases, DEP1 and PT1B, and that are known to cause the dephosphorylation of ERK, so perturb the ERK pathway in this way. So the goal is to identify the substrate, the direct substrate. So phosphatase specificity traditionally has not been considered very high. They've been considered promiscuous enzyme, and in fact, in vitro, they are rather promiscuous. And so you can have a single phosphatase dephosphorylating in vitro many targets. But what's been proven in a variety of cases in vivo that they can show some exquisite specificity. So what are the possibilities? So in specific condition, the substrate recognition is governed by the Michaelis-Menten equation that shows you that if you want to improve specificity, you can do it in two main ways. One is to increase the relative K-capsule Km by changing the shape of the substrate pocket of the enzyme, and the other one is by increasing the relative concentration of the substrate you want to dephosphorylate. And so to measure this in vitro or worse in vivo is very complex on a large scale. We cannot do it. So to get some ideas about substrate prediction, we use a proxy of substrate specificity and increase and the relative concentration. So for substrate specificity, we use a technology that makes use of peptide arrayed on filters or on glass. And while for local concentration, we use distance in the interaction network. So a substrate is considered to be more likely to be a substrate of a phosphatase if it's not too distant in the distantly pro interaction network. For substrate specificity, we use the technology that we developed when we characterize the whole family of SH2 domains. So essentially, we can spot approximately 20,000 peptides covering most of the human phosphotyrosine proteome on a glass slide in triplicate. And this shows that the experiment is reasonably reproducible. The three replicates give the same signal. And I forgot to say that the glass with the phosphotyrosine peptides, approximately 15 amino acid long, are probed with a phosphatase and then revealed with a fluorescent antibody. But there is a problem because phosphatases don't bind stably to their substrate. So to make the experiment possible, we use trapping mutants to turn an enzymatic assay into a binding assay. Trapping mutants are mutants that still recognize their peptide substrate, but are not able to carry the phosphorylation reaction through. So essentially, by changing this cyst into a serine or this aspartate into an alanine, it's possible to have a mutant that still has some specificity, but remains bound to the peptide substrate. So by doing this approach with all our collection of tyrosine phosphatases, we were able to map the specificity of 16 of them. Here you can see the logo of the peptides that are bound by the different phosphatases, and you see that some phosphatases may like to have an aspartate here, some other glutamic acid here and there. So we can map the 16 phosphatases and group them into approximately 16 classes, depending on their specificity. I see that I'm almost running out of time, so I apologize for going a bit quickly through this, but essentially the message I want to tell you is that by doing this experiment, we're able to conclude that phosphatases have some intrinsic enzymatic specificity, but not much. Much lower specificity, for instance, the SH2 domains. So if we plot what is called the information content of the logo, describing the peptide specifically recognized by these two classes of protein, this is the distribution of SH2, while this is the distribution of protein paired phosphatase, with a much lower average information content. So this means that if we want to use this information to predict substrate, we come up with over-prediction. So we cannot do it because we would predict too many substrates. But then we reason if we can combine this feature with another feature, the feature of distance in the protein interaction network. So if a phosphatase binds to a certain protein by some other module of some other adapter, this protein is more likely to be a substrate of the phosphatase than another protein that is sitting somewhere else in the protein interaction network. So we call this weighted interaction distance, and we use this for proxy of local concentration. So to do this, we did some automatic test mining. We look at approximately 3,000 abstracts describing protein interaction with phosphatase. As you probably know, we are running a database of protein interaction, so we have expert curators that look at the abstract and extract the protein interaction information, and we deposit this in our database Mint. So essentially, now we have a phosphatase network linking all the phosphatases not to their substrate, but to proteins that are likely to interact with them. And we can calculate for each phosphatase, for each pair of phosphatase and putative target protein, what we call the width distance. That is the distance in the protein interaction network. So we have two features: ability to recognize by the trapping mutant on the chip a specific peptide sequence, and width distance. So we can combine this and use this to predict substrate, real substrate, not functional target. And I don't know whether you're familiar with this type of graphs that are called clock curves, that are used to give an estimate of how good is a prediction. So essentially, we have a list of proteins that are bona fide subset of the phosphatase, and we look at our prediction method, how well we can reproduce this. If our method would be perfect, we would have a square like this. So if our method would be random, so like picking at random targets, then would be diagonal in this graph. So as you see, in blue I have, in green I have the prediction based on position-specific scoring matrix derived by the chip experiment, and there is some prediction, a good prediction. It's not random, but still not good enough. If we do it simply by looking at width, we get something that is similar. What actually the measure of a prediction is this area under the curve. This is how you see. Both of them is 0.82. But if you now combine it by Bayesian approach, take the two features in consideration, now the prediction becomes much better, and we have an AUC of 0.92. So now we have this method. We can predict the subset. Do we get? Can we get the real in vivo substrate by this informatic approach? So essentially, let's go back to DEP1 and PD1B that functionally are mapped near ERK, and what we were able to predict by the approach that I described to you before, that one should act directly on ERK by dephosphorylating it, while PD1B would act indirectly by dephosphorylating the phosphatase SHP2. And fortunately, I didn't put any gel here to prove that this is correct, because I'm almost running out of time. But just point to this publication by Emanuela Ferrari and Francesca Sacco that actually show that the prediction is correct. So by using this method, we, after mapping functionally the phosphatase in the ERK pathway, we're able to identify the direct substrate. Okay, and I will not discuss this gel. But I'll go directly to the conclusion of this second part, which we have shown that phosphatases have some intrinsic enzymatic specificity. However, if we want to predict substrate, we have to invoke an additional mechanism that can explain the observed in vivo specificity. And then I've shown that distance in protein interaction network can be used to increase reliability in substrate prediction. Finally, systems biology. Yes, I mean, I'm making my living this last year with systems biology, so I cannot come up with a negative conclusion. I think we'll see it in 5 years, whether we have only produced garbage or we have contributed to it. So, but this is trivial. But when you get so much data, perhaps you tend to forget that the data must be good, reproducible. Otherwise, you cannot extract gold from rubbish. And I try to be polite here. The modeling is important. You have different choices, and you have to choose the modeling strategy that you need. And so, following what Einstein said, make everything as simple as possible, but not simpler. I don't talk about statistics, but statistics is important. If you have so much data, what you are doing essentially is something called multiple testing. So this may lead to conclusions that are not statistically solid. So if you have a lot of data, you're likely to end up with something that you believe is important, but then if perhaps it is only because of multiple testing. Finally, there are already a lot of data that are waiting to be interpreted. So before producing a new large data set, follow the advice on the Labio, look what is already available, and think hard before producing more data. So think hard what we are going to do with what we are producing. So these two movie stars are responsible for the project, Francesca Sacco at the top and Federico Berardini, that's more informatically oriented. Unfortunately, Federico left the day before yesterday for Stanford, and Francesca, if she can fix the car that broke yesterday, will soon move to München. But don't worry, we have newcomers in the group, and so I'll be able to tell you something even more interesting in 3 years' time. I have to also to thank the collaboration with Julio Saez Rodriguez in the European Bioinformatics Institute in Kingston, and the screening was done in the facility of Mario Negri-Sud that is led by, with the help of Antonello Venini. Okay, thank you very much.